Hello, everybody. I'm going to talk to you about some of the research that we've been doing. I started out as a postdoctoral fellow with Nancy Spinner, and my project was to do some of the characterizations of the ring. So I think we had about 25 patients from all over the world to define those two groups. And since then, we've been doing some more research on both ring chromosome 20 as well as ring chromosome 14, since these are two chromosomal ring syndromes that both have epilepsy. So since it's quite rare, we were able to get funding and patients who have both syndromes to try to understand what is a ring doing and how is it causing seizures. So as Nancy mentioned before, here are some of the possible reasons why a ring chromosome could result in epilepsy. So the first thing that the thing that we sort of hoped it would be, because it would be an easier answer, was that you are just missing sequences, you're missing genes on that ring chromosome. So by doing our study, we saw that we could rule that out, because most people still had all their genes present on that ring chromosome. So another thing that, one of the other possibilities was that sometimes you lose a ring and you copy that normal chromosome, and now you have two chromosomes, but they're identical to one another, which was uniparental disomy. But I'm thinking we can rule that out too, because recently I was part of a group that found several patients that had maternal UPD. So they had two copies from their mother, and they have a completely different phenotype. They don't have seizures. So I really don't think it's also the uniparental disomy. So that sort of leaves us with, is the ring itself causing seizures somehow by working with, like Nancy showed you, how sometimes you could lose the ring or duplicate the ring. Also this mosaicism for the missing or extra rings, but is the ring also making the cell behave differently? So how does the presence of a ring affect the cell? But also, how does the ring affect the genes on that chromosome? So talking about that transcriptomics. So we also have been doing some experiments to see, does a ring structure affect how our genes are expressed? So the first one, to see how does a ring affect a cell in general, we were looking at cells with ring chromosomes to see, do they grow the same? Do they look the same? Compared to cells that do not have a ring. And then we were also looking at, globally, does the cell express different genes overall? To answer the question of how does a ring affect the chromosome itself, we can use newer technologies to look at expression of genes from that ring itself as compared to genes on the linear or a normal chromosome. So directly comparing a ring to a normal chromosome. So to do these experiments, we really need to know what normal is. Everybody is different, so what is normal? Because what we're going to be really looking for, I think, is a very subtle change. Having a ring is not like a black and white. It's going to affect the cells just slightly. So we needed some tissues to study. So we were lucky enough to get some blood and tissue samples from several patients with both ring 20 and ring 14. The tissues that we are able to get, neuronal tissues are the best ones probably to study, but we can't really get those from people. And we also wanted to look at multiple different rings to see if what we are finding is specific to ring 20, or can it be generalized to any ring chromosome. So first of all, to tackle what is normal. So humans are very diverse. We are not as identical to one another as we think we are sometimes. I'm really into genealogy, so I've been doing my DNA to see where did my ancestors come from. So just looking at our DNA, we're able to break different human populations down. This just shows you how different we really are from one another. So we can't really look at two people and just compare them and assume that the differences are because they have a ring and they don't have a ring. These could just be the normal differences between any two people. There's also differences in the number of genes we have. This is within like 2007, this was the discovery of the year for genetics, that it's normal to have some missing and extra genes. That is perfectly normal. And a person to person, I might have 3 copies of gene where everyone else has 2 copies. So defining what normal is really a challenge. And there's also environmental factors that can affect 2 people. So their age, their gender, what they're exposed to. So in the laboratory, we are studying ring cells from 2 main types of tissues, from blood and from skin, and these are some pictures of the cells in culture. So from cells from blood, we actually have to infect them with a virus, which produces immortalized blood cells that we can grow, not forever, but for a very long time in culture. And then also from skin cells, we're able to grow fibroblasts. So you can see that they look different. So we're really looking at blood cells and skin cells. And these are tissues that we know don't really have a phenotype. There's not a lot of issues with people with ring 20 in these tissues. So this is just looking at one of these LCLs, which is a blood cell line culture. And we started out with a patient who was Mosaic. They had almost 90% ring, and they had about, it was 85%, and so there was 10% that was normal. As over time, this mixture of cells in culture, we saw that ring cells were lost over time, and the normal cells took over the culture. So growing in culture, after about a couple weeks, we didn't really have many ring cells left. So this was very hard then to do any of our experiments, because normal cells just took over. So what we tried to do then is one of the very talented women in our laboratory, Debbie McEldrew, who's very good at tissue culture, she spent a lot of time, and we took patients who were Mosaic for the ring, and she managed to get individual cells from these patients to grow up, cultures that were just the ring or just the normal from the same person. So she did that with a lot of dilution, and we were able to get ring and normal from the same person. And these cells really allowed us to do some of the genetic experiments, because now we didn't really have to worry about the person-to-person difference. From the same person, we had a normal cell and a ring. So we could really use these to see what does the ring do, as compared to what the only difference is literally the presence of a ring. So one of the things that we were seeing with what does a ring do is that we did see that cells that have a ring are different than cells that do not have a ring. They have different sizes, and you can see on the left are the ring cells, and on the right are the non-ring cells, and just visually you can see that the cells do look different from one another. So as we saw before with growing the Mosaic cells, that the cells with the rings just don't grow as much as the cells without the rings. So, and here's with statistics to show that it's in 2 patients we were able to do this with, it was very consistent. So ring cells are just not growing as much. So why are the ring cells not growing as much? There were 2 possibilities that we looked at. Either the ring cells were dying, or the ring cells just didn't divide. So our cells are constantly dividing. So if the normal grows at a certain rate and the ring cells grow much slower, this could also explain why, over time, in our Mosaic cell lines, it just became normal. So I am throwing up the cell cycle, so if people remember this from school. So our cells go through a cycle where they duplicate their DNA, and then they divide and form 2 daughter cells. It's hard to use this. I don't even know how to use this. So here our DNA duplicates, and then our cells divide. And then there's a phase here that the cells just aren't actively dividing. And when we looked at the same person, their ring versus their normal cells, This is showing the distribution of where the cells are in the cell cycle. And here are the ring cells. And you can visually see that there's a difference between right here and here. So mathematically, it showed that the rings are just not dividing. They're not dying. The cells are just often over here in this non-dividing stage. So I don't know how much this actually happens in a living person, but in cell culture, when we have blood cells, the ring cells just don't divide as quickly. So that was just looking at the cells. Well, we wanted to look at the genetics. So we wanted to know, is having the ring affecting the expression? So going back to this concept of DNA, all our cells have the same DNA, but cell types are different because they're expressing those different genes. So we have DNA to RNA. So if you look at the RNA, we can tell which genes are active and which genes are not active. So one of our hypotheses is that the ring is affecting the entire cell. There's many genes on many chromosomes that are affected by the ring. And another hypothesis that's not necessarily exclusive of one another is the ring is specifically affecting genes on chromosome 20. So we used some newer techniques. It was called high-throughput RNA sequencing, where we sequenced all of the genes, all of the RNA that's expressed from these cells. So in our case, we had two conditions, and I got this from a review, so it's not quite the same. But in this case, they were comparing a tumor to normal. And in our case, we were comparing the ring chromosome cells to the cells with two linear chromosomes. And from these cells, we can isolate the RNA. We stick it into equipment that costs hundreds of thousands of dollars. So it's a very expensive equipment to do this. And we were lucky to be able to send it to a facility that could do it for us. And then we were able to see how much of this RNA is produced in the ring cells and compare it to the non-ring cells. So there is a difference between the ring cells and the non-ring cells. And the biggest differences sort of make sense with what we also saw with how the cells grew. That the cells with the ring chromosomes, most of the genes that are differently expressed have to do with cell growth, with cell movement, with their organization and their survival. So this experiment, again, this was done on fibroblast cells, and fibroblasts aren't expressing those critical brain genes, the genes that are only really expressed in the brain that make those tissues function as a brain should. So we're looking at skin. And we know that there wasn't much difference. There's not a lot of skin issues in patients with ring chromosome 20. Another issue with this experiment was we don't know if the differences in cell growth and cell survival are a cause or an effect. So the cells there could be something different with the cells, and the result is that they're not growing as well. So we don't really know why we see this difference, but we are seeing a consistent difference. So one thing that we can really see from this experiment, even though we're not looking at brain cells, is that how is that ring affecting genes that are on that ring chromosome? So in both of these patients, we knew which parental chromosome formed the ring. So we have cells where the same parent's chromosome is a linear and the same parent's chromosome is a ring. So there are differences between all of us that now we're going to take advantage of. The chromosome that came from dad has different sequences than the chromosome that came from mom. So we're able to see the expression of the gene, and we can say this one definitely came from mom's chromosome, and this one definitely came from dad's chromosome. So being able to use these markers, we're going to now see how is this gene, when it's on the ring chromosome, different than the exact same gene on the linear chromosome. So this is just showing the wild type, the normal cell line from one person. And in white and gray are the two different parental chromosomes. And you can see when the chromosome 20 is linear, we have about an equal expression from mom and dad. Each chromosome is producing the same amount of DNA. When we then look at a ring chromosome 20, we can see that the chromosome that's a ring is not producing as much RNA from that chromosome as the linear chromosome. And this is from the same person, just their two cell lines. So having a ring really does affect the amount of genes that are expressed. And it's not just one or two, it's many genes off of the chromosome that are affected. And this is the same data, just plotted in a different way, because a lot of people like to look at this data slightly differently, depending on how you can visualize things. But in this case, we can see this is the ring chromosome, and this is the other chromosome, and this is the same chromosome from the same parent, from the linear chromosome. So you can see that this is different. There's less genes expressed off of that ring chromosome. So that was one of the hypotheses, that the ring is affecting gene expression, and we do see that it is. And again, this is in fibroblasts. This is not in brain tissue. So, I said that already. So what we really wanted to do is look in tissues that we know should have more of an effect, brain tissue. One way that we can actually make brain tissue from other tissues, from blood or from skin, is creating something that's called an induced pluripotent stem cell. This won the Nobel Prize in 2012. So this is a newer technique where you can actually take a blood cell or a skin cell and reprogram it and make it go back to an embryonic state and then differentiate it to any tissue that we can. So we were hoping that we could use this to then take our normal and our ring cells from the same patient and try to make some neuronal cells. And so this is just showing, like, if you start with those induced stem cells, you can make neuronal progenitor cells and then hopefully make neurons. So as Nancy briefly said, that there was another lab studying a completely different kind of chromosomal abnormality, and they showed when they were making these induced pluripotent stem cells that they had a ring chromosome that became monosomy, they lost the ring, and then it duplicated the same chromosome to make uniparental disomy. So before this paper came out, we were also working on it. So we were sort of in parallel. We didn't know this paper existed. But here we are, we're making ring 20 iPS cells. And we were able to successfully make these iPS cells. But when we went to characterize them, to say, okay, how many of them have ring, how many of them are normal, we saw that almost everything became normal again. And so this is what we started out with, 100% ring. And these are all of our different iPS cell lines. And you can see that here's normal in gray. Almost every one of them starts to have normal. And then over time, they became all normal. But it wasn't normal. It was that it copied the same chromosome over. So eventually, in culture, we could not keep the ring stabilized. So we weren't able to do that perfect experiment. However, about a year or two ago, more papers come out because there's newer technologies. And now there is a way of taking a fibroblast cell line and doing a direct conversion. So you can trick the cell, a fibroblast, and it can go right into a neuronal cell without having to go through those intermediate steps. So in this case, here's some cells. And you can go, instead of going through this like pseudo-embryonic stem cell, differentiate it and make the neurons, there's now techniques that you can go right to neurons. So this is what, again, Debbie in our lab, who's very good at cell culture, she was using these newer techniques, And here, this is a ring 14 patient, so not a ring 20, but a ring 14 patient. And these are not fibroblast cells anymore. These are neuronal cells. So this is the stage that we're at right now, is that we are able to make some of the cells. And this is showing some markers to say these actually are acting like neuronal cells. So the next steps are to look at these neuronal models to just make sure that we have made real cells and not some abnormal cell that's not like something you would find in a human, so we have to characterize them. But we can use these now to look at gene expression in cells that are more likely to be affected by the ring and do some functional assays to see how are these cells behaving or how can a ring affect the function of these cells. And so Nancy and one of the collaborators have a grant in order to look at some of these, as Nancy mentioned, organoid bodies, which is a way of having a lot of cells that can interact with one another. We still want to know that why do these rings show these altered gene expressions? There's many different, we know that there's less genes that are being expressed off of the rings, but why? So there's some more detailed questions we can look at. What is causing the expression differences? And maybe if we can get the expression differences back up, that could be something potential in the future. So those are the acknowledgements. You saw them earlier, the same people. This is our team working on Ring 14 and 20.